All right, so this is the May June paper 2023, paper 4.3. Okay, and we are going to move through this paper, go through it, and see all that it wanted. All right. Okay, so let's start with question number 1. It says here, the movement of molecules within an organism can occur by diffusion and active transport. Complete table 1.1 by placing ticks to show the correct features of each process. All right, so here we have the table with the feature, and then we have to tell if it's active transport or if it's diffusion. Okay, so it first says movement of particles always occurs across a cell membrane. Right, so movement of particles occurring across a cell membrane, we have to tell if it's diffusion or active transport. Then we have movement of molecules during gas exchange. We have rate of diffusion of particles is higher when the concentration gradient is larger and requires energy. So movement of particles always occurs across the cell membrane. This is going to be active transport. So we'll take active transport. Okay. And then movement of molecules during gas exchange, this is diffusion, so we'll take diffusion. Rate of movement of particles is higher when the concentration gradient is larger. This is also diffusion, so we'll also take diffusion. And then requires energy from respiration. What is requiring energy from respiration is always. Then explain why active transport is important in root hair cells. All right, so you have to tell why active transport is important in root hair cells. Now we have to know that the concentration of mineral ions, for example, in the roots is high than the concentration of mineral ions in the soil. But the mineral ions are required to move from the soil into the roots. So here it is against concentration gradient. Now, because it's against concentration gradient, obvious active transport has to be applied. Okay. Yes, so when we talk about why active transport is important in root hair cells, we have to say that the concentration is high in roots than soil. Or you can say that movement against concentration gradient. All right, so the movement is against concentration gradient, hence it will require what? Energy. Okay. Yeah, the movement is against concentration gradient, and of course it will absorb the substances from the soil into the roots. All right, let's move further. It says here, number 2, transpiration is the loss of water vapor from leaves of a plant. Complete the sentence describing transpiration. So it says water evaporates from the surfaces of the blank cells into the air spaces and then dash out of the leaves through the stomata as water vapor. So we have to know that water usually evaporates from the mesophyll cells, right? It evaporates from the mesophyll cells into the air spaces, okay? And then from the air spaces, it diffuses out of the leaves through the stomata. So the first blank over here, it will be mesophyll, and then from mesophyll, it will then diffuse out of the leaves. Okay, the mesophyll, it will diffuse out of the leaves. Okay, the next blank says, explain how water moves upwards in the xylem. All right, so water will move from the soil into the roots. All right, so the root hair cell will absorb the water from the soil into the roots. Then from the roots, the water will move into the xylem vessels. Right now, the question is how water moves up in the xylem. What allows or keeps the water moving in the xylem? First of all, we have to know that as water is constantly flowing in the xylem, some of it is being lost as water in form of water vapor, okay, through the stomata. Okay, so this loss of water through the stomata creates something known as transpirational pool. All right, so constant loss of water through transpiration creates transpirational pool, which keeps water moving upward through the xylem vessels. Okay, so the first thing here is going to be transpirational pool. Okay, transpirational pool is the first thing that is allowing water to keep moving. Apart from transpiration pool, we have a force of attraction between water molecules. Right, so the water molecules are held together by a force of attraction that is existing between these water molecules, which is cohesion. All right, so we would say here attraction between water molecules. The attraction between water molecules or cohesion is also going to keep a constant flow of water upward the xylem vessels. But apart from that, we have adhesion. Right? So adhesion, this is like force of attraction between water and xylem walls. So the force of attraction that is existing between the xylem wall and the water molecules will also enable water to keep flowing upwards in the xylem vessels. All right, so we go further. A student investigated the effect of wind speed on the rate of transpiration in a small Vaccaria aquatica tree. They measured the rate of transpiration when the plant was placed in different wind speeds in both the light and dark. The tree was given an adequate supply of water. The results of this investigation are shown in table 2.1. Right, so table 2.1 shows us the results. As we can see, we have a key over here. This straight line over here is light, and this big dotted line is like dark. Then the question says, describe and explain the results shown in figure 2.1. Okay, so these two lines are definitely representing the rate of transpiration. But when you look at it, you'll find that in the light, it appears that the rate of transpiration is high compared to the rate of transpiration in the darkness. All right, that's one thing. But also, for the both of them, it is like rate of transpiration increases with increase in wind speed. Okay, so as the wind speed is increasing per second, the rate of transpiration, amount of water lost by the plant, is also increasing. Okay, yes, so those are the things that we can deduce from this what? From the graph. All right, so we'll write them down. Describe and explain the rate or results shown over here. So we'll go further to say that the rate of transpiration is high when there is light, or in presence of light when there is light, due to the fact that stomata, when light is present, stomata are open. So because stomata are open, more water is diffusing out of the plant. But also the rate of transpiration, increases with increase in wind speed. In presence of light. This is the first thing that we can see. But in darkness, we can also say that the rate of transpiration in the dark is low compared to light. That's also one thing you can see. But also, the rate of transpiration in darkness increases with increase in wind speed. The rate of transpiration in the dark, increases with increase in light, in wind speed, increases with increase in wind speed, increase in wind. Yes. So those are the things that we can see from the graph over there. All right. The investigation was repeated in the light with the same species of tree, in an environment where humidity was higher. Draw a line to predict the results in light with increased humidity. Now you have to know that humidity is one of the factors that affect the rate of transpiration. Okay, along it is like we have light and the wind speed. All right, so when humidity is high, the rate of transpiration usually decreases. All right, yeah, so if it is decreasing and we have to present over on the diagram, on the figure, what will happen? Okay, we just need to put the line beneath the line graph which is presenting over here. It will be beneath rate of transpiration when we have only light, okay? Because when we add humidity as a factor, it will always lower the rate of transpiration. So if it will lower the rate of transpiration, the line that we are going to draw is going to be below this line. So we can project maybe here. Yes. We can project it like that. Okay. The investigation described in 2C was repeated, whereas different wind speeds in both light and the dark. So we have different wind speed. However, the tree was not given an adequate supply of water during the investigation. Predict the results and explain your prediction. Okay, so we are to investigate the high amount of water. Okay, but also the wind speed and light amount are also different. Now, the case is that transpiration is all about loss of water. So if we do not have a high supply of water, the rate of transpiration will not be high. Together, so the rate of transpiration will not be high. So we will say that the rate of transpiration, will be low for both. Will be low for the will be low for light for right phase. When we check rate of transpiration in the right phase, it will be low, right? Why? Because the plant has less amount of water, right? Due to the fact that the plant absorbs less amount of water, but also the stomata close to reduce water loss. Now, due to these factors, we expect the rate of transpiration will definitely be low, right? But also, we see that the rate of transpiration will be low in the dark. Also, will be low in the dark. As tomatoes are closed. All right, so the rate of transpiration in the darkness will change very little. The change will be very small. Why? Because the stomatas are already closed. Okay, move to the next question. Fig 3.1 is a diagram of a section through a human heart. Draw X on a septum. Now, septum is the part of the heart that separates the right side from the left side of the heart. So definitely septum is going to be over here. Okay, explain the reason for the difference between thickness of walls B and C in Fig 3.1. So wall C is the right ventricle and wall B is the left ventricle. Wall B pumps blood with a very high pressure to all parts of the body, while C pumps blood to the lungs. Right. So B is pumping blood to a very long distance. Right. Now, because it is pumping blood to a very long distance, it means it needs to pump blood with a very high pressure. That's why its walls are a bit thicker or a bit muscular in comparison to C. So let's say that B pumps blood to the body. This is a long distance with very high pressure. with very high pressure. All right, yeah. But A pumps blood to the lungs. Yeah. So this is the reason B is thicker than C, because it's pumping blood to the body, which is a long distance with a very high pressure, but A is only pumping blood to the body. Okay, a red blood cell enters the vena cava at A. Explain how the red blood cell is moved from the vena cava to the aorta. Right, so red blood cell enters the vena cava at A, this part over here. Okay, but our question needs us to tell how this blood moves from part A to the aorta, which is definitely somewhere here. Okay, so what is happening is that this red blood cell will be pumped into the right atrium. Okay, then right atrium will pump this blood to the right ventricle. Right ventricle will pump the blood to the lungs for gas exchange. Okay, after gas exchange has taken place into the lung, this blood cell will be carried by the pulmonary vein back to the heart. And when it reaches the heart, it will enter into the left atrium. Right, then the left atrium will pump it into the left ventricle, and eventually the left ventricle will pump it into the aorta. All right, so it will take a path from the right side of the heart to the lungs, then from the lungs back to the left side of the heart. That is the path that is going to be taken. Right, so let me demonstrate here. Explain how the red blood cells move from the vena cava to the aorta. So we'll say that the red blood cell is pumped into the right atrium. Okay, that's the first thing. Then right atrium pumps it or pushes it, pumps it to the right ventricle. Okay, yes. Then we say that it is then carried, carried in pulmonary artery to the lungs. Where gas exchange occurs. Okay. Then from the lungs, it is carried by pulmonary vein back to the heart into left atrium. All right, then left atrium pumps it into left ventricle. Then we'll move from there and say left ventricle pumps it to the body through aorta. And that is how it will move from the vena cava to the aorta. Right. And then here the question says, an athlete measured her heart rate during a running race. She recorded it before the race, during the race, and during her recovery. Her results are shown in figure 3.2. So here are her results, all right? Suggest how the athlete could monitor her activity, the activity of her heart rate. Okay, so she's doing an exercise, and how is she going to monitor her activity? In many ways, right? She can decide to use a smartwatch. Okay, she can listen to opening and closing of valves. She can use the ECG machine. All right, so we can say that listening to closing of valves, closing and opening of valves. That's one way. You can use a smartwatch. Okay, or someone can use an ECG. Okay, calculate the percentage change in heart rate from the maximum heart rate reached during the rest until the rate recorded at 18. So we have to see the maximum heart rate during the rest. Maximum heart rate during the rest is here. All right. Yes. And then to what? Maximum heart rate during the rest, what period? Reached during the rest until at 18 minutes. Okay, so maximum heart rate is here, which is 180 at 15. And after 18 minutes, where is 18 minutes? 18 minutes is approximately here. Here. Okay, so 18 minutes is like, yeah, is like this part here, which can be read as 92. So 18 minutes is here, which is like 92, but maximum is here at 15 minutes, which is like 180. All right, so percentage change in heart rate from the maximum heart rate reached during the race until the heart rate recorded at 18 minutes. So at 18 minutes, when we read it, it's like 92, the heart rate, and then at 15 minutes, which is maximum, it was 180. So in order to obtain percentage change, we'll take reading 1 minus reading 2 divided to reading 1. Whereas the reading 1 is going to be at 15 minutes, which was 180. So to be 180 minus 92 over 180. And then the whole answer is multiplied to 100. So let's see what we will get. $180 minus 92. Oh, reading 2 minus reading 1, sorry. Has to be reading 2 minus reading 1. Reading 2 minus reading 1, which is definitely 92 minus 180. Going to be 92 minus 180. All right, yes. So it's gonna be 92 minus 180. It's negative 88. All right. So negative 88 divided to 180. That one times 100. 48.8. 48.8. Yes, so calculate the percentage. Yeah, so the percentage change is going to be 48.8. Yeah, that is going to be the percentage change. All right. Then we go further to the next question. Explain why heart rate must Explain why heart rate must increase during exercises. All right. So why do we need increase in heart rate when we are doing what? Exercise. Or why is it necessary for heart rates to increase when doing what? Exercises. All right. So here, first of all, as we are exercising, it means we need more energy. Okay. So if we need more energy, it means we need more oxygen that will be used in the process of respiration to give the energy that is going to be used. All right. Okay. So first, heart rate must increase to increase oxygen supply. Okay. To increase oxygen supply. Okay, then when we have high amount of oxygen, what is it happening is that to increase rate of aerobic respiration within the body. Okay, to increase rate of aerobic respiration, and then to provide more energy, that will be needed by the body for the whole process of what? Of exercising, right? Yes, apart from that, heart rate must increase to ensure transportation of carbon dioxide so as it is eliminated. Okay, so increase in heart rate allows for more carbon dioxide to be carried within the blood, so it is removed out of what? Out of the body. Okay, yeah. Then we move further. Urea is a waste product. Describe how urea is formed. Urea is formed by a process called deamination, which takes place in the liver. Okay, so what is deamination? Deamination is defined as the removal of the nitrogen part of amino acids. All right, so when the nitrogen part is removed, what is formed is usually known as urea. So we'll say that it is formed in the liver by a process called deamination. Okay, by a process called deamination. But now, what is deamination? We say that deamination is the removal of the nitrogen part of amino acids. So this is what it means when we say deamination. Okay, state the component of blood that transports urea. So the component of blood that is transporting urea will be blood plasma, because blood plasma is the component of blood that usually transports substances. So this will be plasma. And then state why urea must be excluded. Urea must be excluded because it is toxic. Okay, so urea is toxic, hence it has to be excreted. State the name of the blood vessel that carries blood away from the kidney. So it is carrying blood away from the kidney. This is going to be the renal vein. Okay, renal vein carries blood away from the kidney, while renal artery carries blood to the kidney. And then B says, Fig 4.1 shows a drawing of a nephron in the human kidney and associated blood vessels. Okay, the human kidney together with associated blood vessels. All right, so we have blood vessels and parts of the kidney labeled K, L, M, and N. And then describe how the structures labeled in Fig 4.1 produce urine. So you have to tell how urine is formed from these structures. All right, so the part labeled K, this is the glomerulus. It is found inside the Bowman's capsule. What it does is filtration. So part K filters blood that enters the kidney. All right, yes. So only smaller substances bypass part K to part L. Okay, the substances in part L is known as the proximal tubule. Inside the proximal tubule, there will be selective reabsorption of useful substances such as glucose molecules that bypass part K. These useful substances will be reabsorbed back into the bloodstream. Then the remaining substances, the filtrate, will pass from part L to part M. Inside part M, there will be reabsorption of water and salt molecules. All right, yes. Then after that, the remaining substances will be passed to part N, which is known as the collecting duct, and the substances that collect in N will form something known as urine. So if we are to explain, we'll say that part K filters blood, allowing only small substances, small molecules to pass through. Then these small substances these substances or molecules are selectively reabsorbed, at part L. Okay, part L back into the blood, Okay, yes. Then in part M, is it M? In part M, water and salt molecules or substances are reabsorbed. Okay, the substances that are not reabsorbed at M, collect into N from urine. So simple like that. At part K, which is the glomerulus, filtration is taking place, whereas big substances or big molecules will not pass through, but small substances will pass through. All right, so the small substances that pass through will then go to be selectively reabsorbed at part L, which is the proximal tubule, and after they are selectively reabsorbed at part L, then they will move to part M, which is the nephron, and where we will have a reabsorption of water or salt molecules, and then after that, the remaining substances will collect at part N to form urine. Okay, a farmer applied fertilizer to a field next to a lake. Suggest two precautions the farmer should take when applying fertilizer to reduce the risk of eutrophication occurring in the lake. So in order for the farmer to avoid eutrophication, first of all, they should apply required amount. Right, so if required amount is applied, it means it will be absorbed by the plants. It will not be lost. Apart from that, another thing is do not apply during rain. So when they are applying this fertilizer, they should make sure that it's not rain. So we are on to question number 5, which says, describe the role of progesterone in pregnancy. All right, so progesterone is a hormone that plays a role in females during pregnancy. One of the roles is that it thickens the walls of the uterus, all right, for the implantation of the fetus. But also, it prevents maturation of follicles. All right, so no new follicle is produced at that time. But also, it inhibits luteinizing hormone. So if it is inhibiting luteinizing hormone, it means it is stopping the release of what? Of eggs during the time for ovulation. Those are the roles of what? Of progesterone in pregnancy. So we'll say that it thickens the walls of the uterus. That is one thing that it does. All right, but then again we can say that it stops, or you can say it inhibits maturation of follicles. All right, and then again we can also say that apart from inhibiting maturation of follicles, we say that it inhibits ovulation. All right, so it inhibits luteinizing hormone, or we can say it inhibits ovulation. By stopping luteinizing hormone. Yes, so those are the roles of progesterone in pregnancy. Then we move to number 5b, which says that Fig 5.1 shows a drawing of a section of a human placenta. The arrows show direction of blood flow. So here is the diagram. You can see that we have the arrows in or pointing in or out of the placenta, but we can see we have multiple blood vessels. You have maternal artery, maternal vein, fetal capillary. We can see have umbilical vein, we have umbilical arteries. All right, these are the things that we can see here. All right, so what does the question say? It says substances move across the placenta by diffusion. State the names of two useful substances that move from the blood of the mother to the blood of the uterus. All right, so substances, useful substances that move from the blood of the mother. One is nutrients. Nutrients move from the blood of the mother to the blood of the fetus. All right, so nutrients are so many. We have amino acids, we have fats and many more. And then another important or useful substance that is moving from the mother's blood to the growing fetus is antibodies. All right, yeah, so these are useful substances that move from the mother to the growing fetus. And then using the information in figure 5.1 suggests how the placenta is adapted for efficient diffusion. So we can look at the placenta over here. All right, you see that it has multiple blood vessels: maternal artery, maternal vein, fetal capillary, umbilical arteries. Yeah. So one of the things that make it adapted to its function is dense supply of blood. All right, and why will it have dense supply of blood? It has a lot of blood capillaries, blood vessels over here. All right, yes, so it has dense supply of blood to ensure sufficient supply of useful substance to the fetus, and also killing unwanted substances like wastes from the fetus. So we can say that it has dense supply of blood. All right, so this is one of the features that we find over there. But also it has thin walls. All right, it has thin walls so that to shorten the distance for diffusion. But apart from that, we also know that it has large surface area. Okay, it has large surface area. Okay, so that enough amount of diffusion of gases and useful substances takes place. Okay, the Rhesus factor is a protein that is found on the surface of red blood cells in some people. If the protein is present, then the individual is Rhesus positive. The allele for presence of Rhesus factor is dominant and is represented by letter D. The recessive allele is capital D. Okay, the recessive allele is represented by letter D, which is a small d. If a mother is Rhesus negative and the fetus is Rhesus positive, there can be problems during pregnancy. A Rhesus negative mother and heterozygous Rhesus positive father have a child. Complete the Punnett square and determine the probability of the child having Rhesus positive. So we already know from the explanation that the mother is Rhesus negative, so should be represented by small d's in the area for gametes together. And then the father is heterozygous for Rhesus positive, so obvious the father will have gametes as such. All right, so we have to complete the Punnett square. All right, so one cross that might take place between a mother's gamete and the father's gamete over here will give us this thing. All right, but at the same time, this first gamete of the mother, when it fuses with the second gamete of the father, this is what we will get. But then again, the second gamete of the mother, if it fuses with the first gamete of the father, this is what we will get. And if the second gamete of the mother fuses with the second gamete of the father, this is what we will get. All right, yes, so the probability, complete the Punnett square and determine the probability of a child being Rhesus positive. All right, so for a child to be Rhesus positive, they must have the dominant allele for recessiveness, for positive the dominant allele for Rhesus positive, which is represented by capital D. So if we look on our Punnett square, we have a probability of 2. All right, 2 of the children having the dominant allele for positive Rhesus. So at the end of the day, the probability of having a Rhesus positive child is 0.5. all right, then we move further. Number 6, it says figure 6.1 shows the percentage of land covered by forests in a country from 1660 until 2000. Using the information in figure 6.1, state which 10-year period had the highest increase in the percentage of land covered by forest. Okay, so we can see from our graph here that from 1660, we can see that there was a fall in percent of land covered by forest. But then again, at 1850s, the percent began to increase. All right, so which 10-year period had the highest increase? It is in the 1950s where we see we have a maximum increase over here. All right, so the 10-year period that is being spoken about here is the 1950s. Outline how human activities could cause the change in percentage of land covered by forests from 1660 to 18,000. How human activities can cause change in area that is covered by what? By forest. All right. So we have activities that people do that require them to acquire more land. All right. So one of the activities that people can do that can result into this stuff can include constructions of houses and infrastructure, construction of houses and infrastructures, all right. Other activities can include pollution of the land. People are just dumping stuff anywhere. That reduces the area that is covered by forest. But also we can say farming activities. All right. When people want to farm, they can find themselves that they are going to clear a land so that they get clear the forest so that they get a place to do their farming activities. All right. It is estimated that 40% of plant species are at risk of extinction. Seed banks can store seeds from many species for a long time. Table 6.1 shows some information about one plant species that is stored in a seed bank. There is the table. We can see mass of one seed in gram. We see percentage of oil content. We see percentage of protein content. Now, using the data shown here, calculate the mass of protein. So we know that the mass of protein in one seed is 1.96 from the table. All right. But then again, at the same time, we have been given the percentage of what? Of protein content. All right. Whereas the percentage of protein content, as we see there, it is 11% together. So if we are to find the mass of protein, it means we are going to take 1.96, you multiply it to 11%, all right, which is equivalent to saying that it is 1.96 times 0.11. all right. Okay, so let me work it out here and see what the answer will be. 1.96 times 0.11. all right, so I get 0.2156. all right, so that is going to be the mass of what? Of protein in one seed. And then suggest why many plants can easily be conserved using seed banks. All right, so why many plants can be conserved by using seed banks? First, seeds are small. All right, so if seeds are small, it means that it is easier to do what? To conserve them. It is easier to do what? To maintain them, just because they are small, right? That's one thing. But another thing, it is that seeds can easily, because they are small, you can easily collect. Okay, and if because they are easily collected, it means even storage is easier because they are not occupying such a big what? Area. All right. Yes. So we can say that one of the reasons is seeds are small. All right, but not only that, we say seeds are easy to collect and maintain. So you collect them, you maintain them in just a small area. All right, yes. So these are reasons why seeds can be used. All right, describe why conservation projects such as seed banks are important. Okay, why do we need to have seed banks? All right, or why do we need to store seeds of different species? Okay, all right, so one of the reasons as to why we need to do this is because it helps to maintain the diversity of the species that we are talking about. Okay, we will have the different species of that particular plant. So we'll say that it maintains biodiversity. Okay, yes, maintains biodiversity. Another thing is that it allows for future researches. You know, you have the different species that you have conserved and you need to conduct a research about these species. It will be easier if we actually have such kind of a species. We'll just grow the plant and do the research. So it allows for future researches to be conducted. All right, yeah, that is it. But also it prevents extinction. It prevents extinction of the particular species that we are talking about together. Yes, but also you can say that it helps to maintain vulnerable ecosystems. Not necessarily the plant. We can have ecosystem, right? Like interdependence between species in an environment. So they'll be able to maintain even other kind of species that are depending maybe on that particular plant. So here again we can say that it helps to protect vulnerable ecosystems. Vulnerable ecosystems. Yes, that could be another role that is done by what? By this conservation in form of seed banks. Yes, another thing is that it can helps to protect or to maintain possible drug sources. You might find that lots of drugs that are being produced, they are dependent on a certain kind of plant species. So if these plants are conserved, it means we will have a species that can be used to produce more drugs in the future. So we can say that it helps to protect possible drug sources. All right, it helps to protect possible drug sources. Yes, so let's move to the next question. It says that state the name of the chemical element that is found in all proteins but not in carbohydrates or fats. All right, so the chemical element that is found in carbohydrates and fats and proteins together are carbon, hydrogen, and oxygen. But the one that is only found in proteins and not the other three is nitrogen. And that is what we are going to say here, because this is the only element that is in nitrogen in protein but not the rest. Okay, table 7.1 shows the names of some biological molecules, enzymes, and the organs that produce the enzymes. Complete the table. So the table has a part which has large biological molecule, products of the breakdown of the large biological molecule. We have enzymes that catalyze the breakdown of the large biological molecule and have the organ that produces the enzymes. So where it is missing, we have to fill the specific missing substance. All right, so let's start with the first one, oil. We have to name the products that occur when oil are broken down. The enzyme that catalyzes the breakdown of oil. All right, the organ that produces the enzyme is already named. So oil, when broken down by the enzyme, it forms smaller molecules which are fat acids and glycerol. So we'll say here fat acids and glycerol. All right, and the enzyme that acts on the breakdown of oil is usually lipase. Okay, then we have glycogen. We need to name the products that are formed when glycogen is broken down. So glycogen, when broken down, it forms glucose. All right, yeah, so glucose. Okay, the enzyme is named and the organ that produces the enzyme is also named. Then we have dash, which is broken down to maltose, and the enzyme is amylase. All right, so we can speak of carbohydrates or starch. So here you can name carbohydrates. All right, you can name starch. This one here, when broken down by amylase, they can form maltose. Okay, and the organ that is producing this is the pancreas. Okay. Protein. The enzyme the products that are formed when protein is broken down are usual amino acids. All right, so amino acids. All right, and the enzymes that are acting on proteins to break them down are the proteins. All right, yes, so this break down proteins into amino acids. All right, state two hormones that affect the concentration of glycogen in the liver. All right, so glycogen in the liver can either increase or decrease. All right, so concentration of glycogen in the liver increases when glucose is converted by insulin hormone into glycogen. All right, so the first hormone affecting concentration of glycogen will be what? Insulin hormone. Insulin hormone causes an increase in glycogen concentration in the liver because glucose is being converted into glycogen. So the first hormone will be insulin hormone. And then the other hormone is glucagon hormone. Glucagon hormone brings about the breakdown of glycogen or conversion of glycogen back to glucose. All right, so if it is resulting into conversion of glycogen back to glucose, it means it lowers the concentration of glycogen in the liver. So the other hormone here would be glucagon hormone. All right, yes. Yeah, that was the last question for this paper.